We have Meena Narsimhan, Director of Process Technology at Novilytic, presenting on at-line nanoparticle-based antibody molecular structure analyses. So please welcome Meena.
Good afternoon, everybody, and thank you for coming. You'll see that the title of the talk has been changed. That's because the speaker was also changed. The original speaker was Eric Bowen, but he couldn't come, and that's why I'm here. So to begin, the outline of the talk is on the slide. We will first be talking about coding by molecular recognition, why and how, the features of this technology, some data and application examples, and then future applications, and then I'll be taking questions. So all of us are here, of course, because we work on antibody therapeutics, and all of us know how much it has revolutionized medicine. In fact, a lot of cancer therapies, immunotherapies, and even eye disease, wet macular degeneration, they all have become treatable because of mAbs. But also, all of us know that there's a whole bunch of side effects associated with the use of these mAbs. And if you think about it, it's, if you look at the structure of the mAb is complicated. It's a polymer, and the 3D surface structure is really complicated. There are a lot of contours in the protein. So the function of the mAbs depend on their 3D structure for function, and they function by molecular recognition like any other biological molecules. They have to recognize something in the body in order to function, and if they don't recognize correctly, or they recognize something that's somewhat like what they should recognize, you get these side effects. So we thought, okay, antibodies function by recognition, molecular recognition. So why not develop technologies based on molecular recognition, analytical technologies? And they would tell you a lot about the 3D structure of the molecule, and they could be informative too. So if you look at how mAbs are manufactured, and I think all of us know this too, they are recombinant proteins. They are expressed in mammalian cells, and the cells secrete them into the medium from which they are purified. And because they are recombinant proteins, you can have errors depending on where the gene is localized, the epigenetics, the errors that could happen during transcription and translation, as well as post-translation modifications. The API, the final active pharmaceutical ingredient that you get, is not a homogeneous molecule. It's not a single unique structure. It's a mixture. It has been estimated that one therapeutic mAb could be up to a mixture of about 50 isoforms. They're called proteoforms. And so, recognizing the bad, getting the fraction of the good and the bad actors in this mixture is not a very simple process. It's complicated. Besides that, the growth medium in the broth, they're isolated from the fermentation extracellular fluid of the cells. And this can also have proteins that the cells excrete. They can have metabolites. So you have to almost purify before you do any kind of quality analysis on the mAb. So typically what people do is, they remove the cells from the growth medium, and then at the very least, they do a protein A affinity purification. Very often they do other purifications too, and then they do quality analysis by liquid chromatography or capillary electrophoresis in the simplest case after protein A. Otherwise, the fully pure molecule is subjected at the final stage to enzymatic digestion with trypsin. They analyze the peptides by LC-MS, and then what they get is a laundry list of chemical structures that are present in the molecule, in the population of proteoforms. So these are what they call chemical critical quality attributes. And basically all that they indicate is manufacturing control, manufacturing process control. Because a lot of the post-translation modifications, all that happen as a result of the environment in the fermenter, and that's what the chemical quality attributes would indicate. But if you look at the structure on the left, the modifications that can happen in the mol are distributed throughout, can be distributed throughout the IgG chain, the protein chain. And so how do you know how they affect, and each one of them can affect the 3D structure they can affect the contour of the molecule. So how do you recognize that? So you have to, we posit that a better way of doing the analysis is to look at, to see if the topology, the contours of the molecule are correct, which means there's a good chance that they will biologically function correctly. And this is because, if you think about it, you look at the antigen binding region, which is, which is indicated by the blue, the light blue. So each antigen binding region is composed of six loops. These loops, three of them are in the light chain, 3 of them are in the heavy chain, and they have to function synchronously to recognize the antigen. You can even have a single mutation in any one of these loops that would prevent antigen binding. And because the IgG is a polymer, you can have changes, chemical changes that occur in distal parts of the molecule, somewhere that is totally not near the antigen binding site, but it can affect the antigen binding. You could also have a bunch of chemical modifications that are required to produce an effect. Another bunch of chemical modifications may not have any effect at all. And each proteoform has its own unique set of chemical modifications. So the whole thing becomes very, very complicated. So just having a list of chemical modifications doesn't tell you anything. Which one of these is significant? So if you use fluorophore-coded molecular recognition agent, you can actually compress mAb quality management analytics. For example, if you have a fluorophore labeled Fc ligand that binds to the Fc domain, which is the lower part of the molecule. It's common to all IgGs. You will label all the IgG proteoforms, and it will give you the titer of the mAb. And then, in addition to that, if you have an antigen that is either fluorophore labeled or MASC labeled, and throw it into the analytical mix, you can get double labeling. You get coding of the antigen by one color, fluorophore. You get coding of the Fc region by another colored fluorophore. And if your fluorescence detector can detect both, you get a ratio. You can get a fraction. They actually monitor the percentage of IgG API that is biologically active, that can bind the antigen. Similarly, if you have the FC, there are FC receptors in the IgG molecule that are responsible for activating the immune system of the patient. And if you code that with either fluorophore or MASC, you can measure the Fc receptor binding activity of your IgG. And if you code the post-translation modifications by a chemical reaction, which is kind of a little slower than the molecular recognition assays, you can measure, you can quantify the percentage of IgG molecules that have a given PTM, post-translation modification. So you can actually bypass a lot of purification steps if you use molecular recognition coding. So you take the cell culture, the broth, clarified fermentation broth, and directly by molecular recognition coding, you can get the product's quality attributes, biological quality attributes. And that's because the binding of the fluorophore is so specific to the IgG that the contaminants become more or less invisible. And if there's a low level of non-specific binding, if you incorporate a separation component, you can get rid of all that non-specific binding also, and the whole assay becomes really very specific. So you have, because you have very few sample purification steps, basically remove cells, you have less variability between samples. You also have a lot of time and labor savings. So it's actually kind of cool. So using this, we have a kit on the market called the Proteometer-L. So basically in this kit, you take crude, clarified fermentation broth that contains the mAb or the IgG, human or humanized, and then you just inject it into the column. The mobile phase contains the molecular recognition agent, and the reactor, the column, does both mixing of the analyte with the molecular recognition agent, which facilitates the binding reaction. It also has a separation component. And so you can separate by size the monomer, dimer, and trimer, the aggregates, monomer and the aggregates. So you can get both the titer and the aggregate content. Aggregate content is a very important quality attribute because aggregates are generally immunogenic, and you don't want them. So this technology is Novilytic's patented MASC technology, and the way it works is illustrated over here, basically. And it differs from traditional chromatography in that there are 3 phases. Normally in chromatography, you have the analyte partitions between a stationary phase, which is the solid phase in the column, and the mobile phase. And here, it also has to bind the affinity selector or the molecular recognition agent, which is the 3rd phase. In the Proteometer-L, it works with impure samples, with any LC with a fluorescence detector. And the best of all is no sample purification is necessary. And if you're an analyst, you'll say, that is awesome. Fewer steps. So, and this is just a pictorial example of how it works. So you have the mobile phase containing the fluorescence MASC, low molecular weight MASC reagent. And the crude map, they injected into the column. The mixing and binding happens in the column. And what happens is, because the MAB is 150 kilodaltons, and the MASC reagent is 2 kilodaltons, they move at different linear velocities through the column. And the column itself, because it's turbulent flow, it is a very good mixing agent. And so what you normally do in affinity chromatography or in immunoassays, it takes you half an hour or more to do the mixing, and then you have a lot of washing steps. This column does everything really fast. So, and because of the difference in linear velocity, you can see that the IgG is binding more of the fluorophore, and there's a little band behind it which is kind of white. That is where the fluorophore is depleted from the mobile phase. It's moving slower. And as it passes through the column, they separate more widely, And what you get is a chromatogram that looks like so simple on the right-hand side of the slide. On the left-hand side is if you take crude fermentation broth and just do SEC with UV detection, which is the traditional way, you will find that you get a very complicated chromatogram. You can't really get any kind of estimation of the aggregate content from it, because how do you know where to draw the line? Whereas if you do the Proteometer-L assay, you get a really simple chromatogram. You can see that over here, you see the aggregate peak and the MAb peak, and there's no background at that point from the clarified fermentation broth alone. There is some amount of really low non-specific signal over here. And you'll also notice that, this assay is applicable to any molecule that contains human Fc. It could be a recombinant protein that contains human Fc. It could be any human or humanized IgG, mAb therapeutic, or any reagent too. And because the molecular recognition agent is only 2 kilodaltons and the IgG is 150 kilodaltons, you just don't see any change in the retention time because of the binding. You see also that if you look at the scales over here, it is way more sensitive than doing absorbance because it's fluorescence detection. And this is the trough due to depletion of the molecular recognition agent from the mobile phase. There's a lot of information you can actually get from this chromatogram. The total area under the curve here would be the titer. The relative area of the aggregate to the main peak would give you the aggregate content. Fc fragments, if any, will elute here. They're usually between 25 and 50 kilodaltons if they have Fc. And you see the trough, depending on the speed, the rate at which the mAb binds, you can get a difference in the shape of the trough. This is a fast binder, and that's a slow binder. So depending on how fast your molecular recognition agent binds, you get information, that kind of information from the shape of the trough. Actually, if you extrapolate, you can probably get binding constants too. We haven't done the math yet. This is an example where we did the titer and aggregate assay with biosimilars, with a Rituximab biosimilar, which is IgG1 molecule. You can see that the assay even distinguishes between, this is actually the dimer peak, and this is telling you that there are two types of, at least there's something different in the structure of the dimer itself. There are two types because it's able to see the difference between them. It's based on the hydrodynamic radius, and that's the reason you see it. And you can see that if you you inject different amounts, you get a nice linear curve. And this is, again, a very distinguishing feature of this assay, because if you do, I don't know how many of you have done immunoassays, but if you do a binding assay, even SPR or ELISAs, you get a sigmoidal curve. And in this one, the curve is linear because the analyte is always moving into a fixed concentration of the molecular recognition agent. It cannot fall off. It's just, it only has to go in one way. And this is really easy to quantify anything from a linear curve rather than from a sigmoidal curve. Another advantage of using fluorescence is that the fluorescence detector settings can be varied quite a lot. So you can use it you can change the range of this curve. You use a more sensitive detection, you can go to dilute solutions. You dumb down your detector, you can really use high concentrations. So you can use it at almost any stage of the development process or manufacturing process. Another really nice feature is, because it is, we find that it's the total peak area is a very stable. If you use the same instrument, you can do almost, this is like 600 and something assays we did. There's only 5% difference in the peak area. And this is possible because of that we use a constant amount of the molecular recognition analyte in the mobile phase. And if it's so stable, this assay becomes very amenable to automatic data collection and analysis. And actually, we have a customer who actually thinks that is a very nice feature. And then again, this one I told you already. You can see that, I mean, this kind of thing people have reported in literature, and they say it could be a cross-linked dimer or a monomer association with a fragment. Different papers say it's different things depending on your mAb, I guess. We have done, it works with IgG1, that was IgG1 Rituximab, this is IgG4. Everything looks exactly the same. It works with Denosumab biosimilar, which is an IgG2. And, let me go back. If you notice in all of these, we just bought the biosimilar, formulated it in null clarified fermentation broth of CHO cells. So it works in CHO cell. Any mAb that is made in CHO cells, it works. We have also tested it with HEC 293 cell broth. It works just fine. The linear, if you look at compare the LDRs between NIST mAb, which is the standard IgG1, Rituximab is in, this is the orange. Then you have Denosumab and Nivolumab. The slope is slightly different. This could be because their IgG, it indicating that the structure where the molecular recognition agent is binding is slightly different. The contour is slightly different, and therefore you get a different slope and a different binding affinity. I don't know, we haven't done exhaustive experiments to tell you whether it's universally true that all IgG4s will have the same slope as Nivolumab, all IgG2s will have the same slope as Denosumab. I don't know that. As more and more data comes in, we'll figure that out. And you can see, this was an RE. Research use only Biosimilar because that's easily available. But you can see the differences between two manufacturers. The Originator one is very clean aggregates, and the Biosimilar is different. So you can, for biosimilar developers, it's a nice quick way. Normally if you do protein A purification and then if you want to do titer and aggregate analysis, currently what people do, they do the titer analysis, then they do a protein A purification and determine the aggregate content by SEC UV. And you can't, per sample, it's a minimum of half an hour, if not more. This, you can just take your clarified fermentation broth, 10 minutes, you get your answer. It saves time and labor. We just tested to see, because this is our molecular recognition agent binds to the FC region. We don't know exactly where. So what happens is people make aglycosylated mAbs, and because they're just easier to manufacture. There's at least one on the market that I know of. And will it work? Yes, the assay works. It works. mAbs that contain FC mutations, even in this morning's talks you can see, they are useful for a lot of reasons. One of course, most important being for anti-cancer mAbs. They want FC silent mutations. So this one was an FC silent Rituximab RUO, and it worked fine. It worked for Distelizumab, which is also a PD-1 specific, IgG4. Rituximab is IgG1, by the way. No problem. Assay works for these. And then one of our, this data is from Janssen. They actually took, this is a bispecific antibody that has an FC. And the assay worked perfectly fine. They could actually distinguish between tetramers, timers, and trimers and dimers very well. They got the linear response. And yeah, let me go back. And actually, the aggregate content in their samples was as high as 35%. And we have tested from less than 1%. In some of our samples, it works. So 1%, 35% seems to have a big range, and it still works. The Janssen, they also tested to see if it would work with antibody drug conjugate, which had the degree of substitution was 8. It was crude, unpurified conjugate, and it worked fine. So bottom line, as long as your FC region is intact, it works fine. Or not even intact, it's present for most parts. If there's something is wrong with it, it won't bind, and then you'll know something's really wrong with your mAb anyway. And even the aggregate content, we get asked, I always use this SEC UV method, and you are using a different method. Will I get the same values by your method? That's like asking us, are apples and oranges the same? No, because we are using totally different detection method, right? We're using molecular recognition of FC. And this depends, the fluorescence yield would depend on the environment of the fluor in the molecular recognition agent that is bound to the mAb. And that can vary. And the aggregate, it might be different from the monomer. So, but surprisingly, this was like 76 individual proteometer-Ls we took, and we measured the NIST mAb aggregate content average. It falls smack dab between what NIST reports by 2 methods. They used SEC UV, and they used non-reducing CESDS, and our aggregate content is just in between, which is, I thought, pretty impressive. I don't know why it works, but it works. And yeah. And it's really, really stable. I mean, very reproducible. If you're comparing a whole bunch of clones and you want to find out quickly, in the clone selection or something, which clone is better, this is really, really, really a good way to do it. And in the end, if you compare to some standard method, you will get. The thing is, neither this method nor the traditional method actually measures the mass. It's, the UV absorbance depends on the environment of the tryptophan residues within the molecule. Okay, early signs of, it works in HEC293, like I said. For rabbit mAbs also it works, for rabbit antibodies. It recognizes rabbit IgG-Fc. Then, so we said, okay, it works fine for Titer aggregate. What happens? Can we do, like we were explaining at the beginning, can you actually do simultaneous domain coding? Can you measure, for example, Fcγ receptor binding, which binds here at the hinge region upper CH2, as well as, and get a ratio to find out how much of the Fcγ binding happens in an antibody population? Can you measure antigen binding ability? Theoretically, you can, because you could label the Fcγ receptor ligand by mass or fluorescence, the same way here. And you get two outputs. And if you have, for the bispecific, you have 2 antigens, you can label one by mass, label another one by fluorescence, and then you should be able to distinguish between the different species. And this is data to show that actually you can do it. We tested it. So what we did was we took an Fcγ receptor extracellular domain that is 50 kilodaltons, not labeled, incubated it. The CFB alone is the green line. The FcγR receptor alone is invisible in the proteometer L because it's not fluorescent. NIST-mAb by itself in CFB is the purple peak. When it binds the 50 kD FcγR receptor, the molecular weight changes, and you can see at a molar ratio of 1:2, FcγR receptor to NIST-mAb, you get both peaks. The complex peak and this peak goes down. And then you can see that for a fixed amount of FcγR receptor, if you vary the amount of mAb, you get a linear response. So, theoretically, all these kind of assays have become possible. Another important feature we recognized in our proteometer L is, if you compare the area of the trough, it should be the area equal to the area of the fluorescence area in the mAb, but it is not. This is always almost 2 times the area of that. It doesn't matter whether it's slow binding or fast binding. This is because that means there's signal amplification. You take out a certain amount of fluor, but you're getting 2 times as much signal in the mAb. So it's a signal amplification, and this twofold is, you can actually harness it to do cool things. For example, you can do homogeneous assays without sample purification. You can take a crude sample either from product or process development or downstream process monitoring from a column, just inject it into a T and in buffer without the molecular recognition agent, just as a carrier, and mix it with buffer containing the molecular recognition agent. And the fluorescence reading, you will actually get curves like that. And you can see that they're really fast. In 1 minute, you can determine the titer. It's really, really fast specific. The background, if any, due to contaminants in your crude broth, it doesn't matter. As long as you inject the same volume, only thing it will affect is your y-axis in the plot, in the linear plot. Contaminants, it will affect only this part, because if you keep the injection volume same. And we formulated all the IgG1, IgG4, IgG2, and NIST-mAb, biosimilars, straight line for all of them. Very good reproducibility. And you can also do this instead of having the output from from process development or whatever. You can have the output of a column to do this, and then you will get. You can actually do post-column detection this way. So because of fluorescent, your analyte or proteoforms are separated here. You can do proteoform analysis very easily, and I will show you an example in the next slide. We did charge variant analysis of Rituximab, which was formulated in clarified fermentation broth, and using this method. And you can see the different peaks. This is the main peak. You have a whole bunch of basic peaks. This is the acidic variants. And we did it on 3 days on the same column in 2 different instruments. And as long as the results are very, very good. You actually can even get the titer, because if you inject different amounts, it's a linear response, total area. You get these 2, day 1 and day 3 were on the same LC. And you can see that the retention times are stable. The content of the acidic main and basic peaks, the relative content is very stable irrespective of the LC. And in this case, the retention time depends on the dead volume. That's why between instruments it's varying. Between LCs, it's varying. It's so easy because you don't have to do protein A. You just take crude, inject it. And this is a very, very commonly used critical quality attribute during process development. So the bottom line, you have a lot of applications used for this kind of molecular recognition coding. You have, with a fluorescent molecular recognition agent that binds to the Fc domain of the mAb, it codes it quantitatively. So you can do biosimilar analysis, distinguish between biosimilars and originators, which is very important. You can do aggregation analysis, titer analysis. In fact, aggregation plus titer together. With post-separation detection, we can do charge variant analysis. We can do hydrophobicity analysis. And you can again get both titer as well as charge variant composition. You can analyze ADCs. You can analyze function, antigen binding, Fc receptor binding. And where can it be used? It can be used in product development. You can use it for PTM assessment. We haven't actually done this, but it's on our to-do list, and I see no reason why we can't do it. And so this is our takeaway. We are firm believers that 3D structure via molecular recognition coding must be used in quality management. It's accurate. It doesn't require multiple purification steps. It doesn't require denaturation or disassembly of the molecule. I mean, think about it. If you're doing trypsin digestion and then doing analysis of peptides to qualify a mAb to get the structure, it's like, you take a whole bunch of cars and you try to tell me by a list of the components that they have, this car is better than that car. Can you do it? That makes sense. It's better to do a test drive and find out whether the car works, right? And how good it is. So testing for function, it has virtues. So in our assays use molecular recognition agents to recognize and code 3D structures within intact mAbs in broth without tearing them apart to see if an FDA-approved structural feature of the mAb is missing. The analyses are applicable at all levels of discovery, clone selection, product development, production, purification, and in final product analysis. And end with acknowledgements. Dr. Fred Rainier is our CTO, and my colleagues Mary Bauer, Nathan Morris, Eric Bowen have all contributed to this work, as well as Dr. Harsha Gunawardena of Johnson & Johnson Innovative Medicine. Thank you. I'll take questions, and please feel free to stick around if you want to chat with us. We're available to answer questions.
I don't know if we need a microphone, but since you're not.
Yeah.
Well, maybe I missed it, but what's the binding part of the reagent? Is it lectin or something?
No. Sorry, I'm going to come here closer because I can't hear very well.
Oh, okay, well, sorry.
Could you speak in the mic? I'm just gonna.
Okay, this is awkward. The binding portion of the reagent, what is it? Is it a lectin or a peptide, or is it a secret?
It's confidential. The molecular recognition agent is it's our IP, but it binds to. It doesn't need the glycan portion, Fc glycan, to bind. Right. I know it binds in the Fc region. I don't know where exactly.
It's too small to be
It's 2 kilodaltons.
Yeah, so it's okay. We can all guess.
You can guess. And we've published the paper on this technology in Analytical Chemistry.
Okay. Thank you.
I have one question.
Yeah.
Related to the question, the previous person. So once the knowing the Fc receptor binding affinity in the CH2 region, including hinge. So I guess maybe the production groups are very nervous for the sugar chains located in the CH2 domain. So, the many papers about the sugar moieties influencing on the Fc receptor binding.
Yes.
So have you ever tested your Fc labeling functional analysis for that kind of sugar deficient assay?
Yeah,
Copper.
for the sugar deficient one I showed for A-glycosylated rituximab, it doesn't matter. It works just fine.
Oh.
So whether it's glycosylated or A-glycosylated, no glycan, FcγR, our agent will recognize and bind.
If you have some actuated how these assays function at the working, so
Like what?
so I'm not sure that what kind of peaks or what kind of
Oh, it looks exactly the same, actually. Let me show. Yeah, here. This one was rituximab that contained, it was the asparagine N297. It had a mutation. It was either a Q or an A, N to A. So it cannot be glycosylated. And it looks exactly like, this is a glycosylated one, right? Fc silent, but it's glycosylated. I mean, the same, it looks exactly the same. Actually, if you look, the slope of the LDR between the A-glycosylated and glycosylated rituximab, it was different. But that probably it is recognizing that it is deformed in a certain way, the Fc region. It's different. But as long as you still get a straight line, and the curve looks absolutely fine. It binds.
I think you showed most of the work done with just one fluorophore, right, binding the Fc region. I was curious, you mentioned in the beginning of the talk there is also, you can label the antigen as well. I was curious, can you also add that to this kind of column and then do this biological testing, as you mentioned, where you can test for two fluorophores at the same time?
We did. Actually, we took the extracellular domain of PD-1, which is a very important anti-cancer ligand, and it was labeled with a fluorophore, and you get a fluorescent peak for PD-1, and we used a different color for the molecular recognition agent, so we get a different peak for that. So I was able to quantify it. The problem was, it's. I haven't actually developed an assay that we can sell with that yet. It's all to be done. Yeah.
Thank you. And I am a little bit confused about this results because this method should be used to the kind of CQ critical quality parameter or something.
Yeah.
But people knows that once the sugar chain modified or lost or something, so people knew the FcγR receptor binding or other efficacy will be changed. But the result is the same. So how?
No, because this is only binding to the Fc portion that is unaffected.
Mm-hmm.
If the Fc receptor binding, if you want to measure Fc receptor binding change, you actually have to use Fc receptor as a ligand to measure that change and use this. Yeah, and use this molecular recognition agent to get it to a titer. So you can get a relative proportion. I can say 80% of my product does not bind Fc.
Okay, got it. Thank you.
Okay.
Perfect.
I think there are no more questions. Thank you very much for coming.